Unit 5, Biology, Understanding Genetics to Genetic Engineering. Chapter 9, The Origins of Heredity, Mendel and the Garden Pea Experiment.
I'd like to begin today's lecture by asking a few simple questions. Why do people look different from cats, from horses, and from birds? Why do people look different from each other? Why do you look different from your sister or brother? The answer to all these questions lies in the genes of people and animals. Your genes provide biological instructions that control how you look, how you function, and even partly how you behave. Genes determine how every living creature on Earth appears, how it functions, how it reproduces, and generally how it behaves. Now, genes are found inside the cells of the body and are passed down or inherited from one generation to the next. You may, for example, have inherited the color of your hair from your mother, or your ability to roll your tongue from your father. Or you may have inherited some other interesting trait you may or may not be grateful for from your great-great-great-grandfather or grandmother. Who knows? Inherited traits or abilities are determined by combinations of genes that are different for every human being. Although each individual is really quite unique, it's interesting to note that most genes are common to every person on Earth. In fact, humans share many of the same genes with other kinds or species of animals, from the simple little fruit fly to our close cousin, the apes. Do you realize that the small African ape called the bonobo shares 97% of the same genes we have? I don't know whether that's good news or not for us, or for the bonobos. The study of genes is a branch of the field of biology called genetics, and the scientists who study how genes are inherited and passed from one generation to the next are called geneticists. The field of genetics actually dates back to the mid-1800s, but most of what we know today about genes and about genetic disorders has been learned in only the last few decades. But don't let me jump ahead to what's going on in modern genetics before we acknowledge and briefly examine the work of a pioneer in biology, Gregor Mendel, who lived from 1822 to 1884. Mendel carried out some of the earliest experiments in genetics by studying the simple pea plant. Between 1856 and 1863, he grew more than 28,000 pea plants, and he kept very careful records on these 28,000 plants. From these records, he saw clearly that there was a predictable pattern by which plant traits or characteristics were passed from one generation of the pea plant to the next, a pattern that had never been reported before. He explained the patterns in principles or laws. Today, these principles are known as the Mendelian laws of heredity. Because we're talking about Gregor Mendel, let me make an aside and tell you something interesting about Mendel that might make him seem more real to you. In 1851, he was sent to the University of Vienna to study mathematics and experimental biology, the two subjects that were to make him so famous throughout the world. However, after 2 years of studying at the University of Vienna, he left the university. He was extremely nervous and uptight about taking examinations. It seems that this extreme nervousness about taking examinations kept him from completing his degree. Now, taking examinations makes everyone nervous, but apparently this disability did poor Gregor Mendel in terms of his university studies. However, the examination disability did not stop him from conducting his research or from becoming one of the most famous scientists of all time. Now, where was I? Yes. I was talking about genes and the passing of certain traits and characteristics from one generation to another. Many of your individual traits, including the color and shape of your eyes, the texture of your hair, and even your height and weight, resemble those of your parents. Your parents have passed traits on to you, and this passing of traits from parents to offspring, as we know, is called heredity. From the earliest ages, people have been interested in studying and understanding how heredity works. People have been interested in learning how they could alter crop plants like corn to give corn the traits that would make the corn crop more plentiful and better tasting. And today, they can do that. Humans also have always been interested in learning how to improve the breeding of domestic animals to make them more valuable and useful. In the case of racehorses, for example, owners want to breed strong, beautiful, and fast horses that can run faster than their competitors' horses can run. The famous racehorse Seabiscuit was offspring or son of a very famous champion racehorse called Man of War. Now, undoubtedly, Man of War passed on traits of strength and speed that helped his son Seabiscuit race faster than so many other horses, even though Seabiscuit was a small horse when compared to the other larger horses he raced. Okay, well, where was I? Before DNA and chromosomes were discovered, when Gregor Mendel began his experiments, heredity was one of the greatest mysteries of science. As I mentioned previously, Mendel studied science at the University of Vienna, and there he learned how to use mathematics to try to explain natural phenomena. While in Vienna, he studied the work of a man called Knight, another early pioneer in the study of heredity and genetics. Knight had crossed, or mated, a variety of the garden pea that has violet flowers with a variety that had white flowers. To his surprise, he found that all of the offspring of the crosses had only violet flowers. However, when two of the violet-flowered offspring were crossed or mated, their offspring produced both violet and white flowers. The white color trait had reappeared in the second generation of the plant. Now, Knight was puzzled by this reappearance of the white flowers in the second generation, and he was unable to explain why this had happened. Mendel decided to repeat Knight's work on the garden pea plant, called Pisum sativum in Latin, but he decided to apply his knowledge of mathematics and statistics to help him predict the results, or the outcome, of the crossbreeding of the white flowering and the violet flowering plants. Now, you might ask why Mendel used the simple garden pea rather than a rose or some other flowering plant. Also, why did he use a pea rather than an animal to study the puzzle of heredity? Well, Mendel had a number of good reasons for choosing to study the pea plant. For one thing, the plant was readily available and plentiful where he lived in Austria. Second, the peas could be cultivated quickly and with little effort. And most important, from a mathematician's perspective, the garden pea plant produced a large number of baby pea plants, or offspring. Actually, it seems that the garden pea was a very good subject for studying heredity for several other reasons as well. The garden pea plant has many traits that have two clearly different forms that are easy to tell apart, such as color. Let me give you an example. The color of a pea plant is either violet or white. There is no intermediate form of color. Next, the mating of garden pea flowers can easily be manipulated, so Mendel was able to control the mating of the plant easily and then observe the offspring plants that resulted from the mating. As I mentioned at the beginning of the lecture, Mendel grew more than 28,000 plants and recorded the many offspring of these plants. And he did all this counting and record keeping long before computers were available for record keeping and statistical analyses. Just imagine. We are still benefiting today from the early research done by Gregor Mendel. As a result of his research, today we understand much more about genetics and heredity, though there is still so much to be learned. For instance, we know that genes are tiny biochemical structures inside each cell that determine particular hereditary traits, such as eye color, blood type, maybe intelligence, and so on. But we must remember that the gene only gives the potential for the development of a particular trait. How this potential is achieved or realized depends partly on the interaction of the gene with other genes, and it depends partly on the environment. For example, a person may have a genetic tendency toward being overweight, but the person's actual weight will depend on a number of environmental factors, such as how much and what kinds of food the person eats. Yes, we are what we eat, and thanks to the fact that our ancestors ate and mated, we are. But then it's almost time for lunch, and my inherited hunger trait must be reminding me to end our talk and let you and me go to lunch. I'll see you next time.
Unit 5, Biology, Understanding Genetics to Genetic Engineering. Chapter 10, Genetic engineering in the biotech century. Playing it smart or playing roulette with Mother Nature's designs.
All right, I started out our last session together by asking a few questions that led me to talk about two major issues in biology. One of the major issues I talked about was Mendel's experiments on the garden pea plant and his findings about how certain traits are passed from one generation to the next. The other major issue I talked about was the role heredity and genetics play in who we are, what we look like, and how we generally behave. And I need to stress that word generally when I say how we generally behave. Today, I'll be talking about the topic of genetic engineering. I'll be talking about what genetic engineering is, and I'll be touching on some of the potential advantages and some of the very real dangers that genetic engineering brings with it. I'll be giving some examples of genetic engineering projects, mainly in the field of agriculture, but also in the field of medicine. I'll also mention some of the cons of genetic engineering, especially the new microorganisms. And finally, I hope to finish up the lecture by explaining in more detail exactly how genetic engineering works. I hope to cover all these topics, but as you know, I sometimes get sidetracked or off topic in my lectures, and I sometimes run out of time, but let's see how things go. Okay, let me again start off with some questions. Do you realize that today we can grow orange plants or lemon plants from orange or lemon tissue in vesicles, which are small bladder-like bags? Some industry analysts believe that the day is not far off when orange juice will be grown, as it were, in vats, and there will be no need to plant orange or lemon orchards or groves to obtain orange juice. Question: is this a good idea and a benefit for humankind? Think about it. In the field of animal research, scientists are working to develop engineered super animals. These super animals will have enhanced characteristics for food production, such as extra nutritional value or lower fat content. Some scientists are also creating new kinds of animals, called transgenic animals, that can serve as chemical factories to produce drugs and medicines for other animals, and maybe one day for human beings. Researchers are also trying to create animals that can serve as organ donors for humans. The question: is this the right thing to do to animals? To make them grow to supersize or use them as organ donors for people? That's the question. All right. Here's another possibly more controversial question involving genetic screening in medicine. Should a couple expecting a baby be able to make sure through genetic screening that the child they're expecting does not show signs of having inherited some hereditary disorder, such as very poor eyesight or mental depression, or don't forget that tendency to be overweight? Another more personal question. If you could, would you use genetic screening to find out if your future child or grandchild had an inherited disease in his or her genetic makeup? Now, it's certainly true that many diseases and disorders that people get are caused by agents such as viruses and bacteria, or things in the environment, such as pollutants. But we are learning today that the cause of a number of diseases, like certain kinds of cancer, and a disease like cystic fibrosis, are in fact genetic. These diseases are thought to be passed down from generation to generation. Take the genetic disorder known as hemophilia, for example. Hemophilia is a genetic disorder involving the blood clotting factor. The symptoms of hemophilia are uncontrolled internal and external bleeding if the hemophiliac gets even a small injury. Today, thanks to advances in medicine, the symptoms of hemophilia can be controlled by giving the hemophiliac injections of the blood clotting factor that's missing from the blood. As far as the issue of medical screening for genetic diseases, do you know that in the United States today, hospitals are screening newborn babies for a wide range of congenital or hereditary diseases, such as hemophilia and cystic fibrosis? By the year 2004, every state in the United States was requiring that newborn infants be tested for a long list of inherited diseases. That's the good news. But, and this is a pretty big but, With the knowledge we have of human genetics and with the techniques we have for screening newborns and even embryos, how long will it be before we're screening and maybe selecting human embryos for characteristics such as physical strength or other qualities such as eye or hair color? You name it. Should scientists engage in the genetic screening or engineering of human beings? But let me return to the topic of genetic engineering of plants and animals that's already going on in this biotech century of ours. We have already seen hints of the coming biotech revolution with the cloning of animals, such as the famous Dolly, who was born in 1996. By the way, did you know that Dolly the cloned sheep later gave birth to a lamb, demonstrating that clones originating in the laboratory are able to breed in the traditional way and are able to produce healthy offspring? Now just think about the implications of Dolly for a minute. Today, if we can produce cloned animals like sheep and mice, as we can, are we really that far from seeing the first human clone created? Some people will say, I can't wait for that to happen. Others will be horrified at the very thought. They believe that scientists who attempt to clone human beings, or animals for that matter, have overstepped the ethical boundaries of science and are even endangering the safety of humankind with their cloning experiments. Some people believe that genetic engineering and cloning is playing it smart. Others, however, think that genetic engineering and cloning is playing roulette with Mother Nature. Whatever you may think about the ethics of the genetic engineering of humans or animals, today biologists are using genetic engineering for two main purposes. One, in agriculture, to make hardier plants and crops for agriculture, and two, in medicine, to make newer and, they hope, better drugs for medicine. But let me start off with the field of agriculture. We can see the evidence of genetic engineering of crops sitting right on the supermarket shelves, probably in that box of cereal you bought at the supermarket recently. Did you know that by the year 2004, more than 50 crop plants had been genetically engineered, including potatoes, soybeans, and corn, just to name a few? Can you guess how much farm acreage is now devoted to genetically engineered crop production in the United States alone today? Well, let me tell you. Between the years 1998 to 2003, 68% of the soybean acreage, 26% of the corn acreage, and more than 69% of the cotton acreage were devoted to genetically engineered, or what they sometimes call transgenic crop production. That's a lot of genetic engineering going on in the early years of this coming biotech century, wouldn't you say? You know, I'll bet if you recently ate a tomato, it could have come from a transgenic tomato crop. And I'll also bet you didn't even know that you ate a genetically engineered tomato. For better or for worse, and in many countries, agriculture, especially but not only in the United States, has been impacted in serious and significant ways by the use of genetic engineering practices. The good news is that genetic engineering is able to produce crop plants that are more tolerant of stressful climate conditions, such as drought. In addition, genetic engineering can be used to control how fast or slow fruit ripens, so that crops can be shipped to markets far from the farm that grew the crop. Let me give you another example of one of the potential benefits of genetic engineering of crops. Plant geneticists have developed strains of crops that are resistant to some chewing insects, such as grasshoppers and beetles. The geneticists inserted a certain gene into the crop plants. This inserted gene makes a protein that injures the stomach of the insect that is chewing on the plant. Well, when that grasshopper or the beetle gets a bad stomachache from chewing on the genetically engineered plant, it decides to leave and look for another plant to eat. As a result, the good news is that crops that are resistant to certain insects do not need to be sprayed with pesticides that do tremendous harm to our environment when they get into our rivers and water sources. Now for some bad news about genetic engineering. Let me tell you a frightening story about a gene-altered monster that almost got away. That is, it almost got out of the laboratory. In the early 1990s, a European genetic engineering company was preparing to field test and then commercialize on a major scale a genetically engineered soil bacteria called Klebsiella planticola. For your notes, that's spelled Klebsiella planticola. The genetically altered bacterium was developed to get rid of the crop residues, the part of the plant not harvested for food. The bacteria helped to decompose the plant material. The amazing and frightening thing was that the new soil bacteria, Klebsiella planticola, had been tested, as it turns out, in a careless and very unscientific manner by scientists working for the biotech industry. The biotech scientists believed that the bacterium was safe for the environment and would greatly aid farmers. Well, fortunately, a team of independent scientists decided to run their own tests on the gene-altered Klebsiella planticola. When they did, they discovered that the researchers from the biotech industry had apparently created a biological monster, a genetically engineered microorganism that could kill all terrestrial plants if it were released. After the exposure of the danger of this new bacterium, the gene-altered Klebsiella planticola was never commercialized. My point is that we need to be very careful about testing the safety of genetically engineered organisms before they come to the marketplace. In addition to finding evidence of genetic engineering in agriculture, we can also find evidence of genetic engineering in many of the drugs produced by pharmaceutical companies around the world. A key goal of genetic engineers is to make drugs that can cure genetic disorders such as cystic fibrosis and hemophilia, which I spoke about earlier. Another key goal is to produce new vaccines that will protect people against diseases such as smallpox and malaria, and against new diseases that will arise in the coming biotech century. Okay, I wanted to finish up the lecture by talking about how genetic engineering actually works, but I seem to have run out of time. So I'll ask you to read the section about how genetic engineering works in your textbook. It sums up in a nutshell how the process works. Well, that's about it for today. I'll see you next week.